sort of give you a little bit of an idea of what the type of work that we're doing here at the Guthrie Cardiovascular Research Center. And I appreciate the talk that Dr. Leslie Miller gave you. It really gave you an overview of sort of the diverse cell types that we've been using for stem cell research for the treatment of heart failure. I'm going to particularly focus on induced pluripotent stem cells. Just a brief overview of the talk that I'm going to sort of provide for you guys is basically define what pluripotent stem cells are, what embryonic stem cells are, how we're able to obtain these types of cells, a little bit about the characterizations that we do, and how then we tell these cells to differentiate into the cell type that we want, in our particular case, a cardiomyocyte, and sort of just briefly discuss some of the limitations that we have come across during our work. So pluripotent stem cells are first discovered by, they're sort of embryonic stem cells. They were first discovered by Martin Evans in the mouse embryo, which in around 1981. And so we've been working with mouse embryonic stem cells for about 30 years now. The advent of human embryonic stem cells wasn't until 1996 by Professor Jamie Thompson in Wisconsin. And so sort of these cells, embryonic stem cells in the human, arise from a embryo at the blastula stage. And these are basically inner cell mass cells that are isolated by using either antibodies against the trifecta derm stage, or by laser ablation. So if you ablate this sort of the outside of this blastula, you can isolate these cells, which is what is known as embryonic stem cells, and then these cells sort of have as Professor Leslie Miller told you, the ability to self-renew and the ability to differentiate into really any cell type in the body. And they grow in the sort of the caveat or the advantage of this cell type is that we can grow them indefinitely, so allowing virtually a source of replenishment for directed cell therapies. I'm going to briefly, just in one slide, give you an overview of what so now that we have embryonic stem cells, we wanted to be able to make patient-specific stem cells. And sort of some of this work was inspired by Sir John Gurdon and his lab about using frog embryos to basically reprogram an adult cell into a pluripotent stem cell. And then some of the work that we've done is so a somatic cell nuclear transfer cell is basically something that you can therapeutically clone an adult stem cell nucleus back into a pluripotent stem cell. By removing you can take human oocytes and remove their genome, fuse a somatic cell or fibroblast skin cell nucleus into a stem cell, into these oocytes, and you allow them to develop into embryonic stem cells, basically giving you the opportunity to make patient-specific stem cells. So now basically this cell will have all the inherent genetic components that this cell, in order to make your patient-specific stem cell. There were some difficulties in trying to get human somatic cell nuclear transfer to work. For a long time, people were unable to be able to carry out and follow these cells through the blastocyst stage because they would arrest. And so then Shukrat Milipov basically came out with a protocol where he injects caffeine, allowing you to faithfully reprogram an embryo with an adult nucleus to an embryonic stem cell. A lot of this work inspired basic research in terms of making patient-specific cells from, or induced pluripotent stem cells from other sources. And so Shinya Yamanaka in 2006 discovered that you could take an adult skin fibroblast and reprogram them back into a stem cell, or what we call induced pluripotent stem cells. He was given the Nobel Prize in 2012, and so really it inspired a vast field in stem cell research where you could now look at patient-specific disease models in an induced pluripotent stem cell. You're able to take, say, a patient that has a long QT syndrome, take a 3-millimeter punch hole biopsy, and then be able to grow out the fibroblasts in a tissue culture dish, and then reprogram them back into a stem cell, because then maybe you can now grow them or differentiate them into cardiomyocytes so that you can study the disease pathology. And so basically, again, you're taking fibroblasts, adding these 4 gene factors or cocktails of proteins that sort of govern pluripotency, or what we call stemness. And you can differentiate, you can de-differentiate them into an induced pluripotent stem cell. What this allows you to do then is you can coordinate this iPS cell and tell it to become a cardiomyocyte. Professor Yaqub will talk to you guys about some of the more exciting work that has been ongoing in our laboratory regarding taking an adult skin fibroblast or cardiac fibroblast and directly going towards a cardiomyocyte. My talk will focus mainly on sort of this pathway and how and what we're doing and what we've set up in the laboratory to be able to carry out some of the work that we've been doing. What sort of the first developmental questions that arose was when Yamanaka reprogrammed skin fibroblasts into iPS cells, was that do cells really have a hierarchy? And so sort of the Waddington landscape that a lot of people show is where an embryonic stem cell basically differentiates, and each of these troughs really differentiate between the different types of tissues we have in the body. For instance, a cardiomyocyte would be a different trough than a neuron would take. And sort of once that these cells reach their endpoint, they weren't able to either go back or jump over the hill to become something else. And really, we now know that with the advent of induced pluripotent stem cells is that we really don't. We can go back and then differentiate them into a different type of cell, as well as Professor Yaqub will talk to you about, is basically taking a skin fibroblast and then directly reprogramming them back into a cardiomyocyte. I'm going to briefly jump to the derivation of induced pluripotent stem cells and how we do it in the lab. A couple of years ago, a paper came out describing by fluorescent activated cell sorting or flow cytometry the characteristic protein expression profiles or cell surface markers during reprogramming. So reprogramming, as you might think, is a very long endeavor actually. So to take a skin cell and make it into a induced pluripotent stem cell, it typically takes a little over a month or two to be able to reprogram them back into a stem cell. And when Thorsten Schlager published this paper and mentioned our results suggest that the analysis of the multiple markers is essential to distinguish fully reprogrammed states from partially reprogrammed states, it gave us an impetus to think about if we can isolate these cells using flow cytometry, would it help to distinguish or make better iPS cells versus partially de-differentiated iPS cells. And we published this paper last year, with work that was inspired a little bit in New York and then carried out in Qatar, where we looked at really almost a weekly time point with flow cytometry to look at the pluripotent markers that these cells express on their cell surface. So SSCA4 and TRAL160 are markers that are that are indicative of pluripotent stem cells. And what we saw was that we can follow them after we introduce these 4 genes that control reprogramming. We can follow them over a time scale of 3 weeks and see that slowly these cells turn into what would be indicative of pluripotent stem cells. We can then isolate these cells and sort them out. And we're able to clean up basically the partially differentiated cell types that arise when you reprogram a skin fibroblast into an IPS. We're also, the next question we thought was perhaps we can increase it's a tedious process. As you saw, the numbers are quite low in terms of the efficiency of reprogramming. The efficiency of reprogramming typically lies between 0.01% to 0.1%. And so if you can imagine being able to tediously find these cells under a microscope to be able to pick out these colonies of cells and then expand them is a tedious process. And so perhaps by sorting these cells out, we can decrease the time to, one, decrease the time to make IPS cells, and two, make it with a better efficiency than what we had seen before. And sure enough, we can. And between manually deriving these cells, which typically takes upwards to 2 months to be able to derive, we see essentially there aren't any differences if we just use a fluorescent activated cell sorter to be able to isolate these cells. What I'm showing you here is basically immunocytochemistry of the characterizations that we perform here at the lab at QCRC. And basically, sort of the transcription factor that governs, or is what we call as a gold standard litmus test for determining pluripotency, is NANOG. It's a transcription factor that activates pluripotency, and we clearly see that in each of our manually derived IPS lines, we also have NANOG expression in our FACS-derived IPS lines. TRAL160 again is one of the cell surface markers that we use to sort out IPS cells, and they too are faithfully recapitulated to express in both manually derived and FACS-derived IPS cells. And so what this really allows us to do is really shorten the time scale to be able to make IPS cells from 2 months to almost, we sort in about 7 days, and then in 14 days we pick out the colonies. And so essentially in 2 weeks you're able to make induced pluripotent stem cells, which then you can use to be able to differentiate them into your desired cell type. We also then did extensive studies on karyotyping, and to show that these cells all had a normal karyotype. And then one of the essential things to show is that we can have, we infect these cells with genes that are on viruses, and we want to really show that we can silence the exogenous factors, silence the virus, and that the cell's own repertoire and machinery takes over for endogenous expression of the pluripotency factors. We can then differentiate these cells into the 3 germ layers, and so one of the important demonstrations that we have to show when we make an induced pluripotent stem cell is that are they able to differentiate into any cell type? And so we randomly differentiate these cells into all three germ layers, the ectoderm, the endoderm, and the mesoderm. AFP is an endoderm marker, and we clearly see green. So these cells can differentiate into endoderm. These cells can also differentiate into alpha smooth muscle, which is a mesoderm marker, or sort of the heart lineage. And then also we can make ectoderm by using TUJ1, which is the neurona marker. We can then inject these cells into mice, and and what they go on to form is what's teratoma. And basically a teratoma is a tumor that consists of all three germ layers in it. And so basically telling us that the cells that we've created are indeed induced pluripotent stem cells, because they, one, can self-renew, and two, they can differentiate into any cell type in the human body. Sort of the next thing I'm going to talk to you about is so once we have an induced pluripotent stem cell, we want to be able to direct their lineage specification towards a certain cell type. So how does one go about doing that? And what we've been fortunate enough to do is glean some of the animal studies that people have done in developmental biology to be able to find out what signaling cues are required for telling a cell, a stem cell, to become sequentially restricted towards a certain lineage, in our case, the cardiomyocyte. So early mouse studies essentially showed that certain growth factors and signaling pathways were involved in converting human pluripotent stem cell, or iPS cell, into the mesoderm lineage, which would then often give rise to our mature cardiomyocyte. Most of this knowledge was gleaned from developmental biology, where we saw that DKK1, BMP signaling, Wnt pathways were critical in this specification towards the first heart field, and then the second heart field, and then subsequently the cardiomyocyte. People have been able to make embryonic stem cells or pluripotent stem cells into cardiomyocytes for a number of years, and there's really a number of protocols available that we employ to be able to differentiate pluripotent stem cells into cardiomyocytes. There are, and I'll get to the limitations in terms of where we're at with our differentiation paradigm, and how we can improve that paradigm in order to make a mature cardiomyocyte. But essentially what induced pluripotent stem cells allows you to do is allows you to dedifferentiate an adult stem cell, an adult fibroblast, into an induced pluripotent stem cell, which then you can differentiate sequentially into a cardiomyocyte. And it really allows you to think about sort of some of the applications that it might have. It allows you to have a cell therapeutic application towards it, if you can replace some of the cardiomyocytes that we lose during say a heart attack, or we can use it to study developmental biology. It really gave us the advent of pluripotent stem cells or embryonic stem cells, gave us the ability to study human development over in a petri dish, basically. Before that, we relied heavily on only animal models for studying developmental biology. It also allows us to model diseases in a dish, basically taking either inherited cardiac arrhythmias or inherited heart diseases like hypertrophic cardiomyopathy or long QT syndrome, some of which we've actually begun enrolling patients thanks to the Heart Hospital here and their collaborative effort with Prof. Magdi Yaqub to be able to recruit some of these patients and be able to derive iPS cells from them here at the QCRC lab. And then we're beginning to derive these cell lines and then differentiate them into cardiomyocytes, and hopefully the initial hope is that we can sort of subject some of these cell lines that we've derived in the lab towards pharmacological studies for either toxicity testing or for new drug discovery. We've also thought about how we can direct the differentiation of these cells towards cardiomyocytes. Typically, it's a 30-day process, and what I'm interested in somewhat is how can we tell the cell to become, so this cell, an induced pluripotent stem cell is a cell that can really become anything, and it really has at its disposal a number of decisions that it can make in order to become a mesoderm, an ectoderm, or endoderm lineage. Could we early on help this cell, guide this cell towards making the appropriate decisions, the decisions that they don't become something else, rather than only excluding it into the things that you want them to become. In our case, again, cardiomyocytes. And so we're really focused on, in the lab, is looking at early signaling pathways and how we can manipulate them either genetically or by growth factors in small molecules to be able to direct the differentiation of a pluripotent or stem cell into a cardiomyocyte. We've sort of employed both embryoid body differentiation schemes as well as monolayer differentiation schemes. And so embryoid bodies are basically, literally what you do is you take these cells that are attached on a dish, dissociate them, and allow them to sort of become balls of cells. And then you can add your growth factors or your small molecules or do your genetic manipulations to be able to direct these cells towards different cell types. We've done that in the lab, and if you can see here, we can make induced pluripotent, these are cells that were derived from induced pluripotent stem cells. We differentiate them into cardiomyocytes, and they beat. But perhaps some of the questions that we were also trying to address is maybe you don't want them to beat, because in the adult heart, you have cells that, your left ventricular cardiomyocytes are actually cells that don't beat. They need a pacemaker to beat. And so those are some of the questions that we're going to address. And then also the notion that perhaps when you differentiate these cells, you get a lot of heterogeneity in your tissue culture dish. Basically, you're randomly making ventricular cardiomyocytes, atrial cardiomyocytes, nodal cells. Could you bias these cells and genetically modify some of these cells to give rise to a particular subtype? So we have a couple of ideas that we're going to be exploring in the lab to be able to make atrial cardiomyocytes versus ventricular cardiomyocytes. And what that'll really allow us to do is really then look at some of the different types of disease pathologies. For instance, we don't have an accurate model in terms of the stem cell model for atrial fibrillation. Could we then bias these cells towards atrial cardiomyocytes versus ventricular cardiomyocytes? Once we're able to do that, this is a figure I borrowed from Christine Mummery's group, basically showing that once you're able to differentiate them into cardiomyocytes, you can take iPS cells from a skin patient, from a skin fibroblast with long QT syndrome, differentiate these cells into cardiomyocytes. They don't faithfully recapitulate the disease as you've made the cardiomyocytes, but if you stress them enough with beta-adrenergic stress, you can cause the arrhythmia that you see in long QT syndrome. You can then treat this arrhythmia in this disease model. So basically, essentially, you're able to take iPS cells and make a patient-specific disease model for a particular disorder. And Yamanaka published a recent review on what he thinks, and what I think the field has been moving towards is being able to utilize induced pluripotent stem cell technology to really come up with intuitive ways to be able to use it in personalized medicine. Essentially, you can take these iPS cells and do genome-wide arrays. and glean really multi-omics information, either lipidomics or metabolomics. Use these and see what sort of drugs are particularly suited for a particular patient versus another patient. Because obviously our genetic backgrounds are quite different between patient to patient. We can also, there's also the concept of being able to use it as cellular therapy in cell transplantation. So you can make these iPS cells, differentiate them in cardiomyocytes, and then inject them into a heart either by single cell injections or making a graft and transplanting that graft. Again, I think one of the major important advances of iPS technology is the ability to be able to screen drugs and sort of move the clinical trial process ahead faster, because now you have a in vitro system to test your novel drugs that you would want to use to treat some of the diseases that we're affected with. I want to quickly jump into some of the limitations that we've come across and what sort of ideas that we have in terms of being able to bring induced pluripotent stem cell derived cardiomyocytes into the clinic, as well as just simply using it as a disease model and how we can perfect that disease model so that we can discover new drugs. Some of the limitations in iPS research are that we don't quite fully know faithfully how we can make cardiomyocyte subtypes, like the ventricular or the atrial cardiomyocytes that we're interested in making. Once we're able to differentiate those two, perhaps that might have an insight into the cardiomyocyte maturation that occurs. Could we achieve appropriate maturation of the cardiomyocyte? The other thing that we've worked in, our collaborators actually worked on, is Kathy Liu, who's worked on immunological tolerance. And if we inject these cells into a patient, what actually might happen? We've actually have some data from mouse studies that if you take an embryonic stem cell derived from a particular mouse and you inject them back into a mice, you have immune tolerance. But if you take an iPS cell from that same mice, from an iPS derived from this mice, reprogram them back into an iPS cell, and then inject them back into a mice, you get immune rejection. Something is happening epigenetically that isn't entirely explaining why we get this immune rejection when we're really using autologous cells for transplantation. And then the other question that sort of arises is the arithmogenicity of these cells. That when we make these cardiomyocytes, they beat on their own, and if we transplant them in the context of a mouse heart or a rat heart, we aren't able to control some of their pacing mechanisms entirely. So one of this is a recent paper published by Andre Terzic and Timothy Nelson's group out at the Mayo Clinic, where they so when you differentiate these iPS cells into cardiomyocytes, you basically get cardiomyocytes, but you also get this heterogeneous cell population. And what they've done quite elegantly is they've targeted DNA topoisomerase 2 with a drug, etoposide, that kills cardiac progenitor cells, or allows you to get rid of some of the dividing cells in the heart. And then they transplant these into a post-MI mice. And so you basically, so typically when you would, if you were to inject these cells into mice, iPS-derived cardiomyocytes, there's a heterogeneous population of cells, and you get tumors forming in the site of injection. if you treat them with something that will kill the dividing cells, you can alleviate some of the tumorigenicity that we see in the transplant. And really, this comes back to perhaps we can better understand if we better understand the process of maturation and subtype specificity, could we then eliminate heterogeneity that way? The other limitation about cell therapy and using embryonic stem cells or even induced pluripotent stem cells is there's a very elegant paper in Nature by Charles Murray out at the University of Washington, in where he's taken these cardiomyocytes and grafted them into monkeys. He states clearly our previous studies in mice, rats, and guinea pigs gave no evidence of arrhythmias after human embryonic stem cell cardiomyocyte engraftment, whereas here we consistently observed arrhythmias. And sort of, again, could we make better cardiomyocytes and mature them properly in order to get rid of some of the arrhythmia complications we get? Again, the QCRC approach is we've got Dr. Christophe Renaud and Christian Bohlensdorf and many others that are working on several possible mechanisms to alleviate the arrhythmias and either transplanting these cells with sort of a pacing mechanism or being able to maturate the cells. So in conclusion, iPS cells are extremely important for both regeneration and as a disease model. And at QCRC, our focus is on strategies for both induction, derivation, and differentiation is being perfected. Several remaining challenges need to be addressed. Among them are immune tolerance and cardiomyocyte maturation. And thank you for allowing me to sort of give you a brief overview of some of the IPS work that we're doing at QCRC.